Hey gang, and welcome back to another exciting episode of Biology Online. And today we're going to talk a lot about DNA. Now, in the first part of the notes, we actually covered DNA replication, some of the structure and some of its function. And that's going to be used for two different videos that are available online for you to go through and fill in the notes. The videos are by the amazing Amoeba sisters, and they do such a great job of explaining DNA and DNA replication that I didn't see any point in reinventing the wheel. So the worksheet or the notes for the first part are actually covered in those two videos. So watch those videos and be sure to fill in the notes as you go. So this next part will be a quick little lecture about DNA transcription and translation. So actually going from the blueprint for building something, which is DNA, to actually building the thing that the DNA tells us to make. So you probably remember that a few years ago, it seems like a few years ago to me, but it's probably more like 25 years ago, a wonderful movie came out called Jurassic Park, and more recently we had Jurassic World. And the idea behind these movies was that some scientists had gotten together and said, hey, inside of this amber is an insect that probably fed on dinosaur blood, and that's all been preserved for millions of years. So maybe we could drill into that amber and get the preserved dinosaur DNA out. And they do. And in the movie, they explain they do a really good job of explaining exactly how the process is for all the pseudoscience, for actually going in and extracting all of this stuff and making the dinosaur essentially. But along that way, they use some information called Mr. DNA, right? And he comes through and he says, I'm found in every single drop of your blood. And he does the same thing, but he explains, hey, you can go from the DNA which is preserved to actually taking the DNA which is broken down some, and they used amphibian or reptile DNA to fill in the gaps in the genetic code and complete the code. And then the movie says, and from there, we make a baby dinosaur. So my question is, how do you actually go from the blueprint to the actual physical object that the blueprint tells you to make? How do you go from the DNA to the actual baby? Well, they didn't need to explain it in the video. We already know how that happens. That is transcription and that is translation. So think about it. If you had a blueprint for a house and you wanted to build a house, how would you go through and do this? Well, you would have construction workers, right? And you'd have to have all the supplies. But you have to have workers to come through and read the blueprints and build it. And there's very specific jobs that these different construction people have. You have plumbers, you have electricians, you have people who do the drywall, who do the painting, who pour the foundation, who do the roofing. I mean, everything is very specific, and it has to be done in a specific order. And all this is guided by the blueprint. And turns out that when you have DNA and you want to make a living thing, well, what would you need there? Who are the blueprint? Like, what is the blueprint? Who's going to read it? Who's actually going to bring in the parts? And all this is explained in transcription and in translation. So we call transcription translation, we call this the central dogma of biology. And it's the idea that DNA codes not directly to a protein, but it codes to RNA. And then that RNA codes for the protein itself. And so the first part of this, going from DNA to RNA, this is transcription. The second part, going from RNA to a protein, that is translation. So in the bigger picture, when we do DNA replication, we're just making more DNA. We're making more of the blueprints. So transcription and translation is taking the blueprint and making what it says to make. In this case, it tells us to we make a copy of RNA from it, and then that RNA copy tells us the order of amino acids we need to make the protein. And then that protein that forms an enzyme most of the time, and it goes through and does stuff in our body, like hooking monomers together to form a polymer, like lipids or carbohydrates or the other biomolecules. And those form the cells and form the organelles, and those cells make up living things and form who and what we are. And that is the central dogma of biology. So in the big picture, where is this happening? Well, transcription takes place inside of the nucleus, whereas translation takes place outside of the nucleus. So if you think about it, do you ever want to take your DNA outside of the nucleus? And the answer is really going to be no. There's too much that can go wrong with the DNA. Mistakes can happen. There's a lot of stuff going on outside in the cytoplasm. You don't want your only copy for that cell, the only copy of its DNA, to get messed up. So the DNA never leaves the nucleus. So how do we get the message out? How do we get the information in the DNA out? Well, we transcribe it into RNA. And this type it's a special type of RNA called messenger RNA, or mRNA. And we don't send all of the messages. We just send the specific part coding for the enzyme or the protein or whatever it is we need specifically to make at that point in time. So we transcribe from DNA to RNA, and then the RNA leaves and goes outside in the cytoplasm, where then it's read by a ribosome in the process of translation, and is turned from the information from on the messenger RNA is coded for a specific sequence of amino acids. Those amino acids form a protein. They'll fold up and make a protein or an enzyme or whatever else you need to make, and there you have it. You've gone from the blueprint for something to something physical that's capable of moving and doing things in our body and helping to build us. So transcription inside of the nucleus and translation is outside in the cytoplasm. So how would this happen? Well, here's a DNA strand. All right, so we're going to do transcription. I haven't put the bases on there just for clarity's sake, right? So we have our primed ends, and so we need to just make a specific length of DNA that's going to be copied. So we've got to know, first of all, where to start. So there's this area called the promoter. And it tells the enzymes where to start making the RNA copy. The same thing goes with where to stop. How do we know where to stop? Where there's a specific region of DNA. Usually it's rich in thymines and adenines. Often we call these the Tata boxes. And it basically tells it where to stop. And essentially it either codes for something that ends the process by not binding anymore, or it just basically is a nonsense region where the genetic code doesn't code for anything. So it's not really copied. And in DNA replication, we use DNA polymerase to make more DNA. So what do you think we're going to use when we make an RNA copy? Well, we're going to use RNA polymerase. So remember, scientists are lazy, right? DNA polymerase, RNA polymerase. So RNA polymerase hooks onto the promoter, to the region where it says, hey, this is where you start, and it starts moving down, and it starts making a strand of RNA from the original strand of DNA. And again, this is so that we can take this message out into the cytoplasm. We don't have to bring the entire strand of DNA with us. So it stops and reaches the terminator, and it breaks off. Now, if this needed to be copied multiple times, there'd be another RNA polymerase right behind the first one, and so forth and so on, making as many copies as we needed. And generally, it's a lot. We usually need a lot of copies. Well, this messenger RNA is now ready to go off into the cytoplasm. So here's a little animation of that same process happening. You can see we start the promoter, run to the terminator, and we make our strand of RNA. Now what happens when it hits the terminator? Well, it derails the polymerase, and there we're done. And that floats off again into the cytoplasm. And again, the process repeats as often as we need to for whatever enzymes or proteins that we need to make for the different cells and the different functions in our body. So now we're ready for translation. We're going to take this RNA, and we're going to go from nucleic acids to the language of proteins. So from nucleic acids to from nucleotides rather to amino acids. That's why we call this translation. It's kind of considered like two completely different languages, because before we're going from DNA to RNA. That's still nucleic acids, still doing with nucleotides, but now we're going from nucleotides to proteins. So nucleotides to amino acids. So whenever we read a strand of messenger RNA, when I say we, it's actually going to be a ribosome that does this. It reads it in groups of 3, and we call this a codon, right? So a codon is a group of 3 bases found on the RNA, and it's read by a ribosome. So the ribosome, also called rRNA or ribosomal RNA, is 2 small units, and they hook together, and when they do, they crunch in on and pinch that messenger RNA right in between them. And this way they can now read it. So it reads, again, 1 codon at a time. That first codon is going to match up with what we call an anticodon, which is found on tRNA, which stands for transfer RNA. We call it transfer because it brings in an amino acid with it. So AUG would match up with what? Well, the A would bind with a U, the U would bind with an A, and the G would bind with a C. So we're looking for UAC. And if you look on here, oh, well, there it is. It's methionine. So methionine is going to bind to this. And when it does, it brings in, when this transfer RNA comes in, it brings in the methionine, the amino acid with it. Well, okay, we're moving on to the next one now. The next codon would be CUG. CUG would code for GAC. So where is that on here? Oh, there it is. Okay, it's alanine. And now at this point we have two amino acids next to each other. So the ribosome does something really kind of neat. It forms a bond between the two amino acids. That's called a peptide bond, if you remember back from our macromolecules. And so now it moves on to the next. What's going to bind with GGG? Well, that would be CCC. It's going to bring in an amino acid with it, and it's going to form a peptide bond. Notice that when the ribosome moves down and it detaches from the tRNA, the tRNA floats off, but because the amino acid is now binded to each other, the amino acid stays behind. So we start forming a chain in this fashion down the length of the messenger RNA until we get to the end and we get what's called a stop codon. Right, this codes for not an amino acid, so there's no bond made. So when the ribosome keeps going, it just the amino acid is now free to float off. Hopefully we've made it correctly. We put all the correct amino acids in the right order, and we can now we've now made a protein. Right, so the ribosomes detach, break off into their subunits. Another ribosome can run down the same strand of messenger RNA and make another copy of this. After they do this, usually there's one ribosome right behind the first one. There'll be a third one right behind the second, and they are all running down, making their copies of the making the messenger RNA into the chain of amino acids. So if we did this correctly, that chain of amino acids will fold up and form a protein that's going to do something cool in our body, like help be like catalase and help break down hydrogen peroxide into water and oxygen, or maybe it'll do something cool like be a digestive enzyme and help break down starches that we eat into simple sugars, so that way we can process them. Or maybe it'll do something like build lipids, or maybe it'll make more proteins that we need for our body. Who knows? There's all kinds of options and available things out there that it can do, and that's great, and that's what we need, and that's what we like to see. So here's a little animation of that happening. Just the mRNA is read by the ribosome, brings it in. I did say it was a little animation, right? So it's really short. But that's how we go from a genetic code in DNA to actually something that's real, that's got tangible structure to it, that's coded for in the DNA, something like that Parasaurolophus, the dinosaurs at the bottom of the screen. It's all made this way. You're made this way. I'm made this way. Mosquitoes are made this way. Your teacher's made that way. All that fun stuff. So now scientists use this code, this chart, to help determine what amino acid is going to be brought in by the codon. So we can kind of skip the tRNA as far as when we're trying to figure out what's going on. So we read this in groups of three. So the first one is AUG. So the first base up here is an A. So it's somewhere in this row. Okay. The second base is a U. Here's U under the second basis. So where does this row and this column meet up? Well, it's in this block. And the first amino, and AG is the last one in this row. Here's the last letter. So it's MET. So the first amino acid would be MET. The second one, CUG. Here's C's. Here's U's. So somewhere in here. G is the last one, but you can see that all of these code for the same amino acid, LEU. And we use this code in that way to be able to help determine what amino acid is going to be brought in based on the messenger RNA. On your exam, I'll have this chart available for you, and I'll give you a section of messenger RNA, and I'm going to ask you to translate the RNA to an amino acid. So that's going to be fun. Get ready to look for that. But this is the chart that we use, and you keep going until you got to a stop codon. Okay, so in the movie Jurassic Park and Jurassic World, it's really cool to look at all those neat little dinosaurs and what they're doing, but remember, they're made from, yes, they were taken from amber, the mosquitoes, the DNA, all that fun stuff. We filled in the gaps, filled in the codes, but then we use transcription and translation to go from the genetic code and the blueprint to actually making a living, breathing organism. And so remember that when you go to your exam, be sure to study so you can be a clever girl. Right? So if you have any questions, be sure to email me or message me or contact me or come by and see me if you're on campus, and I'll be happy to help you out with any of the information. And until then, I hope you have a good, nice, warm weekend. A little cold here today. And I look forward to seeing you again next time. Bye.